Welcome everyone. So my name is Antje Kremer and I'm working as a global product manager at Miltenyi Biotec. As part of the regenerative medicine team, I'm managing the hematopoietic stem cell product portfolio. And today I'm very happy to talk about human hematopoietic stem cell research and how to make it smart and easy. This is the disclaimer of my presentation. At first, I will give you a short introduction, which leads us to the topic of today, the human hematopoietic stem cell HSC research. For human HSC research, we are going to talk about the isolation, the culture, and the analysis of human HSCs. So let's start. What are stem cells? All stem cells share two main properties, which are self-renewal and potency. Self-renewal leads to cells that have the same properties as the mother cell. So in a very easy way to say, stem cell renewal describes a process by which stem cells divide to make more stem cells. Potency is the ability to differentiate into a more specialized cell. This can either be a cell of a certain tissue or a stem cell, with less potential than the mother stem cell. The level of potency defines the type of stem cell. According to the stem cell differentiation ability, there are different levels of potency. Totipotency is the ability of a single cell to divide and produce all of the differentiated cells in an organism. One example for a totipotent stem cell is the zygote, which is formed by the fertilization event. In contrast, pluripotent stem cells can only differentiate into embryonic cells. So, pluripotency refers to a stem cell that has the potential to differentiate into all cell types that derive from the three embryonic germ layers, the endoderm, the mesoderm, or the ectoderm. Based on their origin, pluripotent stem cells can be classified as embryonic stem cells, short ESCs, or induced pluripotent stem cells, short iPSCs. Multipotencies describe stem cells with the potential to differentiate into discrete cell types. Multipotent stem cells are, for example, mesenchymal stem cells, short MSCs, or hematopoietic stem cells, short HSCs. Today, we are going to learn more about human HSCs as multipotent stem cells. HSCs are tissue-specific adult stem cells, which can differentiate into all blood cell types. Simply, the process of blood cell formation is called hematopoiesis. It occurs in the hematopoietic system, which includes organs and tissues such as the bone marrow, the liver, and the spleen. The first signs of hematopoiesis occur during embryonic development in blood islands within the yolk sac and in the aortic gonad mesonephros region. These early HSCs migrate in later developmental stages into the placenta and the fetal liver, which serve as the main compartments of prenatal hematopoiesis. Shortly before birth, HSCs migrate to bone marrow, where adult hematopoiesis takes place throughout life. Every day, the adult hematopoiesis must balance an enormous production, as the average person produces more than 500 billion of blood cells. HSCs are found typically in the bone marrow, especially in the pelvis, femur, and sternum. Besides bone marrow, HSCs can be found and isolated from blood sources, such as umbilical cord blood, buffy coat, peripheral blood, or apheresis products. Depending on the starting material, the HSC content varies. Apheresis refers to the process of separating cellular and soluble components of blood. It is a non-surgical procedure. That means blood is drawn from one arm and passes through a machine that collects the stem cells. The remaining portion of the blood is then returned in the other arm. For mobilized apheresis, drugs such as tevacrestim mobilizes stem cells from the bone marrow into the peripheral bloodstream before the apheresis procedure takes place. By mobilized apheresis, it is possible to reach higher numbers of HSCs, approximately up to 2%. For the HSC researcher, the typical workflow looks like this. Routinely, HSC-containing samples are processed over density gradient centrifugation to pre-enrich mononuclear cells as starting material for subsequent isolation of HSCs. For the culture and expansion of HSCs, it is important to add cytokines to maintain the differentiation potential. For the analysis of HSCs, specific markers are used to define the phenotype, and HSC colony assays are used for functional analysis. At Miltenyi, we provide products for the complete workflow of HSCs. By going now through the workflow, I will give you more information on each topic. Let's start now how to make your isolation of HSCs smart and easy. First, let's have a look on some important markers of HSCs. Engraftment analysis of HSC populations using xenograft models revealed that the majority of cells migrating into the recipient's bone marrow express the surface marker CD34. Furthermore, these cells lack expression of lineage-specific markers, show no or very low expression levels of CD38, and express markers such as CD133 and CD117. For the positive isolation of HSCs, the most widely known marker is CD34. It is highly expressed on HSCs and to a lower extent on hematopoietic progenitors and early committed cells. Today, I would like to focus the CD34 enrichment by our MAX technology using the CD34 microbeads. Our MAX technology is a column-based technology for immunomagnetic cell separation, which is based on antibodies coupled to magnetic beads. The nano-sized superparamagnetic MAX microbeads. Our specific MAX microbeads are available for the positive selection of numerous cell types and cell subsets. Here, the positive selection for CD34 plus cells is shown. Positive selection means that a particular target cell type is magnetically labeled. In the first step, the CD34 microbeads are incubated with the cell suspension. During incubation, the antibody bead complex binds to cells expressing the corresponding epitope CD34. During separation, in the second step, the column is placed in the magnetic field of the MAX separator. Then, magnetically labeled CD34 plus cells are retained within the column, whereas unlabeled cells flow through. After a washing step, the column is removed from the magnetic field and the target cells are eluted from the column. This is shown in step 3. The separation process can be automated by using our Automax Pro instrument. This automation provides a high level of standardization and thus ensures reliable and user-independent results. Our MAX microbeads are biodegradable and non-toxic. As already mentioned, the MAX microbeads are conjugated to highly specific antibodies against a certain cell surface antigen. They bind specifically to cells expressing this antigen, and thus preventing non-specific labeling. Due to the low labeling concentrations and their small size, MAX microbeads do not lead to activation of target cells. By these scanning electronic microscopic images, you can see the proof that our column-based MAX technology is gentle to cells. Here, on the left side, there is no visible labeling on the cell surface after cell isolation with MAX microbeads and MAX columns. However, here on the right side, cells were isolated by a column-free technology from another manufacturer. Here, you can see excessive labeling indicated by the white arrows, and this was becoming obvious after isolation. Due to the gentle and effective nature of the MAX technology, the isolated cell populations of interest are highly viable and pure. These isolated cells are compatible with any downstream application. For the direct CD34 enrichment by MAX technology, I want to introduce two of our products to you. The CD34 microbead kit allows positive selection of human HSCs and progenitor cells from various starting materials. Starting materials include peripheral blood, umbilical cord blood, apheresis harvest, bone marrow, and differentiated embryonic stem cells and differentiated induced pluripotent stem cells. Those HSC-containing samples are first processed over density gradient centrifugation to pre-enrich mononuclear cells as starting material for subsequent CD34 enrichment. The same starting materials are applicable for our CD34 microbead kit UltraPure. These microbeads have been particularly optimized for the use with samples that contain large amounts of cell debris or low numbers of target cells. They greatly improve recovery and purity of the sorted populations by specifically enriching viable target cells. And the CD34 microbead kit UltraPure is as easy to use as the classic CD34 microbead kit. Here, the starting material was peripheral blood mononuclear cells, short PBMCs. The separation was performed either with the CD34 microbead kit on the top or the CD34 microbead kit UltraPure on the bottom. Shown is the positive fraction of the PBMC sample after separation. The comparison of the forward to side scatter plot demonstrates that the classic method, the CD34 microbead kit, cannot avoid the carryover dead cells or debris when these are present in the original sample in large amounts. The special formulation of the CD34 microbead kit UltraPure, however, avoids this debris carryover and results in a much cleaner fraction after separation, as you see here on the bottom. Here you see an example for CD34 separation from a bone marrow sample. The separation was again performed either with the CD34 microbead kit on the top or the CD34 microbead kit UltraPure on the bottom. In general, CD34 plus frequencies are higher in bone marrow than in PBMCs. Due to this higher starting frequency, debris carryover is in general less of a problem with bone marrow, and the difference between our classic and the UltraPure kit is less pronounced. However, the data demonstrate that you can expect the same results as you have seen from the PBMC sample. Again, the CD34 microbead kit UltraPure avoids debris carryover and results in a cleaner cell population after separation. Here, an example of CD34 separation from cord blood is shown. The separation was again performed either with the CD34 microbead kit on the top or the CD34 microbead kit UltraPure on the bottom. Again, the comparison of the classic CD34 microbead kit and the CD34 microbead kit UltraPure demonstrates the superiority of the CD34 microbead kit UltraPure with regards to less debris carryover as shown by the cleaner cell population after CD34 enrichment. So, please keep in mind for HSC isolation. CD34 is a well-established marker for HSCs. Our CD34 microbeads allow positive selection of human HSCs and progenitor cells from various starting material. The two introduced products allow an easy one-step isolation of CD34 plus cells. The CD34 microbead kit UltraPure is the ideal choice for low-frequency and debris-rich samples, as it greatly reduces excess of dead cells and debris carryover. The CD34 microbead kit UltraPure is as easy to use as the classic CD34 microbead kit and also suited for all routine applications. After isolation, the HSCs needs to be cultured. For the culture and expansion of HSCs, it is important to add cytokines to maintain the differentiation potential. So let's have a look what is the smartest way to culture human HSCs. The number of HSCs in human samples is often insufficient for downstream applications. HSCs can be expanded in culture with HSC-relevant growth factors to obtain higher cell numbers or to study mechanisms and signaling pathways driving self-renewal or differentiation. Early-acting cytokines such as the stem cell factor, short SCF, FLT3 ligand, and thrombopoietin, short TPO, expand primitive HSCs. Although current HSC culture systems only allow limited expansion and maintenance of primitive HSCs, that are able to engraft and completely restore the entire blood system. Our StemMax HSC expansion system ensures robust expansion of human HSCs. It consists of the StemMax HSC expansion media, XF, and the StemMax HSC expansion cocktail. The StemMax HSC expansion media is a xeno and serum-free medium specifically formulated for HSCs. The StemMax HSC expansion cocktail is an optimized set of cytokines which can be added to the medium to create a xeno-free culture environment with high expansion rates of HSCs. Our StemMax HSC expansion system ensures high levels of CD34+ cells with a broad differentiation potential and the characteristic surface phenotype of early HSCs. Let's have a closer look at the expansion rate and the CD34+ cell count. In this experiment, CD34+ cells were isolated from cryopreserved cord blood using the CD34 microbead kit. Then the CD34+ cells were expanded for 7 days in StemMax HSC expansion media or in various commercially available alternative media. All media were supplemented with the StemMax HSC expansion cocktail. On the left side, you see the fold expansion of CD34+ cells, which was measured by flow cytometry. Our StemMax HSC expansion system achieved an approximately 20-fold expansion. On the right side, the CD34+ cell content is shown. For visualization, the results were normalized to the StemMax HSC expansion system. When looking at the CD34+ cell content after expansion, the StemMax HSC expansion system performed consistently among the best HSC culture systems. Thus, our StemMax HSC expansion system provides high CD34+ cell content. The cord blood-derived CD34+ cells were also analyzed for the differentiation potential after 7 days of expansion. Cells cultured in the StemMax HSC expansion system maintain a broad hematopoietic differentiation potential, giving rise to all major colony types, including primitive CFU gems. Furthermore, the expanded cells were analyzed for the surface markers such as CD34 and CD133 by flow cytometry. The expanded cells in the StemMax HSC expansion system maintained their characteristic surface phenotype of early hematopoietic progenitors as shown here on the right side. Taken together, our StemMax HSC expansion system ensures high levels of CD34+ cells with a broad differentiation potential and a characteristic surface phenotype of early hematopoietic progenitors. For the analysis of HSCs, specific markers are used to define the phenotype, and HSC colony assays are used for functional analysis. So let's see how to make your analysis of HSCs smart and easy. HSCs are routinely analyzed by flow cytometry based on various extracellular and intracellular markers. We at Miltenyi Biotec offer a wide range of unique and standard monoclonal antibodies for research of HSCs and progenitor cells, including CD34, CD38, CD133, and so on. Our reaffinity recombinant antibodies do not require Fc receptor blocking. Additionally, they all have the same isotype, and therefore less isotype controls are required. This saves time and effort. Our Biodye family of fluorochromes enables multicolor flow cytometric analysis with high fluorescence intensities and low spillover. Our Release fluorochrome technology enables the removal of any label after cell sorting. Second, I would like to shift your attention to the functional analysis of HSCs. This is performed by HSC colony forming unit assay, short HSC CFU assay. HSC CFU assays help to examine the ability of HSCs to proliferate and differentiate into colonies in response to cytokine stimulation. This is performed in vitro. In the HSC CFU assay, cells are cultured in a culture medium supplemented with growth factors that drive differentiation into the myeloid lineage, resulting in the outgrowth of distinct colonies, the colony-forming units, short CFUs. Each CFU develops from a single founder cell. Thus, the differentiation potential of the founder cell will determine the colony type that develops. For the conventional HSC CFU assay, cells are cultured in semi-solid medium. This semi-solid medium is based on methylcellulose supplemented with FBS, BSA, and different growth factors. Our StemMax HSC CFU media are designed to maximize growth and differentiation of progenitor cells and allow the single cell to grow in a distinct colony. This is performed in a 6-well plate or 35 mm dishes. After culture for approximately 14 days, the different colony types can be classified by color and morphology using a stereoscope or an inverted microscope. This requires training and expert knowledge. For example, a CFU gem has a compact area that is usually central to a peripheral flat lawn of translucent cells that may be either large or small. Therefore, it is also known as the fried egg appearance. A BFUE is typically found as 3 to 8 densely packed clusters. The color of hemoglobin-containing cells ranges from dark red-orange to brownish. A CFU-M is a sparsely growing and flat colony consisting of less than 20 translucent large cells. A CFU-G is a flat colony consisting of 20 to 40 translucent small cells with a germinative center. A CFU-GM is again a flat colony consisting of 20 to 50 translucent small and large cells. So, the identification of the different colony types based on visual scoring via microscopy is not that easy, right? Today, I want to introduce you our remarkable user-independent HSC CFU assay, as it takes this guesswork out of your conventional HSC CFU assay. The user-independent StemMax HSC CFU assay kit combines differentiation in methylcellulose-free medium with a flow cytometric readout providing clear results. The assay is performed in round-bottom 96 well plates in a liquid culture environment. The medium of the StemMax HSC CFU assay kit corresponds to the composition of methylcellulose-based HSC CFU medium. However, as mentioned, it does not contain methylcellulose. After culture for approximately 14 days, the different colony types are then analyzed by flow cytometry with specific markers. This flow cytometric readout is used for colony counting and identification of the different colony types. As example, any colony which exhibits at least 15% of CD15 positive, 15% of CD14 positive, and 20% of CD235A positive events would be a CFU gem. All other colony types are identified in a similar way. So how does the experimental setup look for the StemMax HSC CFU assay kit? First, the cells are diluted in StemMax HSC CFU assay media and then plated in a round bottom 96 well plate. The StemMax HSC CFU assay media is supplemented with growth factors as the conventional media, which drive the differentiation into the myeloid lineage, resulting in colony formation. So after proliferation and differentiation for 14 days, the generated colonies are stained with the StemMax HSC CFU assay cocktail, followed by flow cytometry. For the flow cytometric analysis, the first step is the gating strategy for the performed staining for CD15, CD14, and CD235A. First, all acquired cells need to be gated, as shown here in A. For that, use a forward scatter versus side scatter plot to set a gate that excludes debris and includes all cells. Then, open the events of this gate on a new plot, as shown here in B, and set a new gate that displays only single events. Then you have to display the single cells in two new plots. For that, find a well with CD235A positive events. On the first plot in C, adjust the y-axis to the CD15 APC channel and the x-axis to the CD235A PE channel. On the second plot in C, adjust the y-axis to the CD15 APC channel and the x-axis to CD14 Bio-Blue channel. Afterwards, you have to set a quadrant to separate the populations as shown here in D. Once the gates are set, the same gates can be applied for all samples. The most important information on these plots are the percentages of CD14, CD15, and CD235A positive events, which are displayed on each quadrant. According to the percentages, the colony type is identified. So how are the different colony types identified? Here again, you see the two already mentioned plots. The plot on the top shows the CD235A versus CD15, and the one on the bottom, CD14 versus CD15. As mentioned, the most important information on these plots are the percentages of CD14, CD15, and CD235A positive events. If over 50% of positive events are measured for CD235A, it is a BFUE colony. By getting more than 50% of positive events for CD15, it is a CFUG colony. A CFUM colony shows over 50% of positive events for CD14. Any well which exhibits at least 30% positive events for CD14 and 30% for CD15 would be a CFUGM. A CFUGEM is identified if a well exhibits at least 20% of positive events for CD235A, 15% for CD15, and 15% for CD14. Here, I show you some exemplary plots for the different colony types. The percentages of CD15, CD14, and CD235A positive events are displayed on each quadrant. The BFUE shows CD235A positive events, at least 50%. The CFUG shows CD15 positive events, again, at least 50%. The CFUM shows CD14 positive events, also at least 50%. The CFUGM exhibits at least 30% positive events for CD15 and 30% for CD14. Finally, the CFUGEM has at least 15% positive events for CD15, 15% for CD14, and 20% for CD235A. So taken together, the colony type is identified according to the percentages of CD15, CD14, and the CD235A positive events. The message percentages of each marker are exported in an Excel sheet. This can be used for straightforward colony identification. The user-independent StemMax HSC CFU assay kit is comparable to the conventional assay. Here, we compared the results achieved by a StemMax HSC CFU assay kit based on flow cytometric analysis with the conventional assay based on visual scoring. Here, CD34 plus cells were isolated from three different sources: mobilized leukapheresis, cord blood, and peripheral blood mononuclear cells, PBMCs. The CD34 plus cells were seeded in the media from the StemMax HSC CFU assay kit or the conventional assay. After incubation for 14 days, the generated colonies of the conventional assay were analyzed by visual scoring. The generated colonies of the user-independent StemMax HSC CFU assay kit were stained and then analyzed with the MaxQuant. As you see here, the distribution of the different colony types is highly comparable between both methods. Thus, the user-independent StemMax HSC CFU assay kit is comparable to the conventional assay. Taken together, the StemMax HSC CFU assay kit combines an easy-to-handle methylcellulose-free medium and a highly standardized analysis. Compared to the conventional assay, which is based on visual scoring, the StemMax HSC CFU assay kit allows a flow cytometric readout, providing clear and user-independent results. The analysis can be automated by using the MaxQuant and thus allows for high throughput measurement. Here, a short summary for the functional analysis by HSC CFU assays. The conventional HSC CFU assay is performed in a 6-well plate or 35 mm dishes. The cells are cultured in a semi-solid culture environment, which restricts cell movement and ensures clonogenic growth. The colonies are analyzed by visual scoring. Compared to the conventional assay, the user-independent StemMax HSC CFU assay kit is performed in round-bottom 96-well plates in a liquid environment. After proliferation and differentiation, the generated colonies are stained and analyzed by flow cytometry. The flow cytometric readout is user-independent and provides clear results. As repetition, these two plots possess a high CD235A positive staining, as you see here. And as we just learned, this is then a BFUE. Easy, right? With that, I would like to summarize. First, we had a look at the isolation of HSCs by CD34 enrichment using our MAX technology. We have learned that the CD34 microbead kit Ultra Pure is the ideal choice for low-frequency and debris-rich samples. It is as easy to use as the classic CD34 microbead kit and also suited for all routine applications. Second, we talked about how to culture HSCs in a smart and easy way. We have learned that the StemMax HSC expansion system ensures high levels of CD34 plus cells with a broad differentiation potential and a characteristic surface phenotype of early HSCs. Finally, we had a look at the analysis of HSCs. After talking about the flow cytometric characterization of the HSC phenotype, we focused on the functional analysis. Besides the conventional assay, I presented the StemMax HSC CFU assay kit, which convinces with easy handling, a user-independent flow cytometric readout, and clear results. Moreover, the flow cytometric analysis is amendable to automation when using the MaxQuant instrument. With that, I'm coming to the end of my presentation. Thank you very much for your attention.